Okay, so again, the godfathers. Here's what we can do is let's go down. So this gene list, if we measured it directly, there's going to be a number here. We did measure CD38 directly. Here is the type of molecule it is, whether the software thinks based on the targets, its known targets, whether it's activated. Here's the confidence in that activation. And you can think of this as standard deviation above or below random noise. So the more positive this number is, the more confidence that this software has that interleukin-4 is more active. Here are all of the targets that we found in the gene list that are associated with interleukin-4. The probability of getting 71 targets in our gene list is 1.2 times 10 to the negative 17. Even though I don't measure interleukin-4 directly, I know it's there. You can drop say you throw a stone into a pond, I might not see where you threw the stone, but I see the waves, the ripples that stone leaves, and I can follow those waves back and find the original stone. Totally. I tell this story to everybody. It's a pretty funny story. So I was analyzing a data set. This was a clinical trial, and there was a bunch of elderly men, and I think they were getting a pulmonary hypertension disease, clinical drug. And doctors are notorious for using the worst controls. So they had their clinical group, and they go, oh, we need the controls, and they just started getting a bunch of people, right? So I got some other old guys, and they could be on all kinds of stuff except the clinical drug. So I did the analysis, basically I was looking in the bloodstream looking for effects of the clinical drug on genes. I didn't really see much of a clinical effect from that drug, but I found another effect. What do you think that was between the two groups? What do you think? It's drug. Elderly men. Testosterone actually is Viagra. Vasodilator. Yeah. Viagra was the biggest response between those two groups because the clinical group couldn't take it anymore, right? That was a great lab meeting to go in there and tell this investigating group put so much effort in their clinical drug, hey, your drug got overshadowed by boner pills. There's nothing I can do. That was tough. But if basically anything that leaves a mark in the transcriptome, I can see. If it affects drugs and we know what kind of genes it affects, I will see it here. It can be drugs, it can be microRNAs. I think this is one of the best ways. What happened, Harry?
Oh, 7-inning stretch.
Oh, there we go. That's the thing with these drugs. Okay, I gotta stop talking about this, but I can see drugs. So let's go down to STAT3. You'll see it kind of down here. You'll see a 3009 down here. Select that row. Again, I know STAT3 is probably playing a role. I read their paper. Now I want to get my own, basically customized STAT3 network. So go display as a network. If you look, STAT3 is in the middle here. All of these genes on the outside of it are genes in our gene list that changed in our activated T cells. These are part of these 747 genes. If it's green, it means it went down in the activated. If it's red, it was increased in the activated compared to the germinal. The odds of getting all of these genes associated with STAT3 by random chance are incredibly small. Therefore, I know STAT3 is playing a role in my system. Number one. That's the first thing. STAT3 is there. Now I got to figure out what STAT3 is doing. The software knows based on the scientific research, and you can click on any of these lines and it'll get you to that reference. STAT3 is known to induce IRF4 and all of the ones with an orange line. And sure enough, they are all going up. STAT3 is also known to inhibit these two genes, NRP2 and this histocompatibility thing. They are going down. The only genes that don't follow a more active STAT3 state are the ones in the yellow. Given the fact there's a lot more orange and blue lines than yellow lines, the software is very confident that STAT3 is up. That's as simple as it gets. That's what it does. And you can do this for anything. If you know these relationships, you can test for anything there. And if you think about it in cancer, that's huge. Is that you got hormones, blood vessels, you've got all these things in your bloodstream that you will never find the source of that are impacting your system. I might not see where it's coming from, but I know what's doing to my system and I can find it. One other thing, and this is the cool thing I want to show you here. This is where you're getting to your own pathways. Again, we got more genes associated with STAT3 than that stupid GO pathway they gave us. Click on STAT3. I'm going to show you why, I love IPA. This function here kind of makes it worth everything. Go to build. So it'll be on this top bar here. Go to build. What I want you to do is I want you to pick grow. Okay, this window should come up. Go down to general settings. I want to find anything that has a direct connection to STAT3. But here's the difference. I've already seen its targets. I want to get all the molecules I can. But here's the deal is I don't want to see anything downstream. I just want to see upstream. I want to figure out what makes STAT3 so active. Right? This is part of the story. If I can find out what's maybe affecting it upstream, then I can affect its function. And then here's the deal is I want to limit my molecules just for the genes that I put in. So those are the only genes it can pick from. I could also use another gene list, say, of genes known to be mutated in lymphoid B cell. How cool would that be, right? You've got something we know is affected at the transcript state, and then you can tie that to a mutation. That's huge. Hit apply. Science! Again, CD44 is here. We've got MET. Again, what we're doing is we're looking for things that are overrepresented that are probably either stimulating this. This might be an inhibitor, and it's going down. So if I'm looking for things to modulate STAT3, these are the things that I'm looking at. Now here's where I'm going to do. I'm going to put some kinetics on this. Here's what I want you to do is I want you to select all of this. Let's draw a big box around everything. I've got everything radiating out of STAT3, and that's great and all, but that's not how things work, right? It's not a bunch of pipelines, it's more spider webs. Now what I'm going to do is I'm going to figure out the spider webs here. So I've selected everything, hit build. Now go to your third one down, connect. I want to see direct connections. So here's what I want the software to do is all of those genes that I know are connected to STAT3, I want you to connect them if you can. If there's some evidence of a relationship in the biological literature. Hit apply. He blinded me with science. Yeah. This is pretty much what STAT3 is really doing in your system. And it is common sense. If I'm looking for big time players for STAT3, I'm looking for things either downstream or upstream that are connected to a lot of things on our list. Like CAV1, CD44, MET. I think that's MET, yep. This is the pipeline. We could even display this as subcellular. We can look at where are these things located at. Look at all this stuff in the cell membrane. Holy, oh my gosh. Wow. If you're looking for things, transcription factors, you would look down here. Again, I want to stress, bioinformatics is not a pipeline. It's not a kit. We've become too much kit scientists. That everything is just, you just throw a kit at it. Go get that kit. Get this kit. This isn't a kit. You have to be on top of these algorithms to make it work. And I'm going to leave you with one story that I think kind of illustrates this. So everybody knows that IBM's Big Blue, the supercomputer, can beat the world's greatest chess player, right? Did everybody know that? No one knew that? Okay, they can. Here's what they don't tell you is that chess player can then get a computer and beat Big Blue. There is something about the human and the computer together that makes both better than each. You have to be on top of this software. Do not allow the computer or let the computer give you an answer. Okay? You have to be there. All right, any questions? Everybody's like, way too much information, man. Honestly, play around with this. You guys have this for a month. You got it for an extra week. Honestly, I give this to my high school kids and they just go to town. Don't worry. You're not going to break anything. This is all web-based. There's no rules. If it works, it worked. What this is for you to do is to help you. It can give you preliminary data. It can tell you where you might want to, what targets you might want to look at next, all of this stuff. If you have any questions, email me if for some reason you're having a hard time getting access.
Thank you.
Yep, no problem, guys. Thanks.
Okay.